Good morning or afternoon to everybody. My name is Neil Halsey, and today we're going to go through the review of the types of vaccines that are available, how these vaccines are made, and the administration of vaccines. First, we need to define what we're talking about. If you use a medical dictionary's definition, it's a bit stiff. Attenuated or killed microorganisms or proteins that are derived from them that are administered for the prevention, treatment, or amelioration of infectious diseases. I think a much more practical definition that I use is a product that is administered to induce an immune response for prevention or treatment of disease. Vaccines have been used since the late 1700s, and actually the first person to administer what we now consider to be a vaccine is a farmer in England by the name of Benjamin Jesty. He noted that he was protected against smallpox after having had cowpox lesions on his hands that he had acquired from a cow. He subsequently inoculated his wife and 2 children with cowpox and demonstrated that his child was subsequently protected against a challenge with smallpox with a process called variolation 15 years later in 1789. Edward Jenner is given credit for the application of the best science to prove that this process, in fact, did work. He was aware in the early 1790s of rumors in the community that milk maidens who had had cowpox lesions on their hand were later protected against smallpox. So he undertook a systematic study of this by, in a very public way, first inoculating a boy by the name of James Phipps, and in those days they published all the names of everybody, which we don't do today. This 8-year-old boy received cowpox on the arm, and the cowpox had been obtained from a milkmaiden named Sarah Nelms. She acquired it from a cow named Blossom. Jenner subsequently deliberately exposed Phipps to smallpox through variolization, which takes the smallpox lesions from the individual who's affected and inoculating it into another arm. Phipps remained entirely well. He then repeated this process, as he quotes, on a number of children, and he coined the term vaccination. Smallpox vaccine, or vaccinia, the virus we're using today, is called a xenotropic vaccine. This is a virus taken from another animal species and then administered to humans for prevention of a related but not identical infection. This is a natural means of attenuation because the virus isn't adapted to the human species. Now we know today this doesn't always work in terms of attenuating or making the virus weaker. If you just imagine what we know about SARS, that's a virus that's adapted to another animal species, but when infecting humans causes very severe disease. So we were lucky with smallpox. It wasn't until more than 150 years later that viruses were grown in tissue culture, and John Enders, Tom Weller, and Fred Robbins won the Nobel Prize for this major accomplishment, which opened the door to growing not just vaccinia, but not all other viruses, but many other viruses that we have today, and the opportunity to make vaccines against them. The process for attenuating most viral vaccines is through serial passage in tissue culture. Measles vaccine virus was isolated from a boy named David Edmonston over in Silver Spring, Maryland, by John Enders and their colleagues, and they passaged that in various tissue culture lines, as are abbreviated here with 2-letter initials, and you can see 24 and then 28 passages, so many passages involved, and that has led to the development of multiple measles vaccines. Zagreb vaccine, Moreton, Edmondson B, and Schwartz vaccines and AIKC were all made through this process, but they're different vaccines because they were serial passed through different cell lines. So the first methods that were used were attenuation, either through the use of a xenotropic vaccine, serial passage, or now today we have a process called reassortant vaccines, where they take the genetic material from a wild-type virus, incorporate it into a parent virus that has already been attenuated, and administer that. That is the way the live attenuated intranasal influenza vaccine, or FluMist, is developed. The live attenuated vaccines that we have today include the smallpox vaccine, BCG, or vaccine against tuberculosis, measles, mumps, rubella, varicella, or chickenpox, zoster, oral polio vaccine, intranasal influenza vaccine, rotavirus vaccine, yellow fever, and Salmonella typhi, or typhoid fever vaccine. The next method to be developed was inactivation, either through heat or, much more commonly and more successfully, through the use of formaldehyde or formalin. That's the way that Jonas Salk developed inactivated polio vaccine from the viruses that had been identified by John Enders and his colleagues. Other examples of inactivated whole organism vaccines are the whole cell pertussis vaccine and hepatitis A vaccines that are now widely used in this country. One can take an inactivated vaccine and then break apart the organism through ultrasound and other techniques and purification methods to use only a portion of the organism, usually the outer shell or antigens that are expressed on the outer surface, because that's what the human immune system sees. And we know from many different organisms those are protective antigens. Examples of these subcomponent or subunit vaccines are the meningococcal and pneumococcal plain polysaccharide vaccines. All the influenza vaccines used in the United States are subunit vaccines. Toxoids are made from toxins. They're proteins or enzymes that are produced by bacteria, and then they're inactivated with formaldehyde. Diphtheria, tetanus, and pertussis are all three different toxins that are combined to make DTaP vaccine. The pertussis vaccine does contain some other subunit components, which we don't have time to review here now. Some of the purified subcomponents of organisms, such as the outer polysaccharide coating on many bacteria, are not very immunogenic, especially in very young children. Through the process of chemically linking those polysaccharides to a protein, one can develop a conjugate vaccine and make the antigen much more immunogenic. The three conjugate vaccines that we have in widespread use today include the Haemophilus influenza, or Hib, vaccine, pneumococcal conjugate vaccine, and meningococcal conjugate vaccine. These three vaccines protect against the most common forms of bacterial pneumonia, meningitis, and sepsis that used to cause a great deal of disease throughout the United States, and now these vaccines are being introduced into other countries where they will also prevent against these severe forms of disease in very young children. One can take the genetic material, the DNA, from an organism, insert selective parts of that into a yeast or a bacteria, and induce that yeast or bacteria to express an antigen, serve as a factory, if you will, to produce the purified antigen, and then purify it and get what's called a recombinant vaccine. If you think about it, this is a mechanism for growing up a portion of the organism without ever having the intact organism around, and this would be very attractive for things like SARS, where you wouldn't want to be trying to grow that virus in your laboratory, as we've already seen a couple of examples of where the virus got out and infected laboratory workers, and with a high case fatality rate. The examples of these recombinant vaccines that are in use today include the hepatitis B vaccines and the human papillomavirus vaccines. All of these vaccines are currently made through this recombinant technology. All of them also use brewer's yeast as the primary factory for production of the vaccine antigens. One can take the actual naked DNA and insert it or inject it in special techniques. It turns out that that DNA then induces the host cells to induce or produce some of the proteins that might be involved in protection against disease. This process looked to be very promising based upon numerous studies in animals, especially mice, And experimental hepatitis B, HIV, malaria, and cytomegalovirus vaccines have been made through just what's called naked DNA. Although they work well in mice, none of these are working very well in humans today, and it's not clear that it's going to pan out or that we'll ever have a licensed naked DNA vaccine. If we do, I hope they change the name, because I think most of us know that some members of the public would be a little uncomfortable if you said you're going to inject naked DNA into them. One can also take that isolated DNA and insert it into another live organism, a bacteria or more commonly a virus, to get what's called a vectored vaccine, so that the other organism, which may be very benign and cause no disease, can carry with it the genetic material to express proteins or other antigens that can induce protection. Examples of vectored vaccines include experimental tuberculosis vaccines using either adenovirus or the modified vaccinia ankara as a vector, malaria vaccines using adenovirus as a vector. We have had experimental HIV vaccines, but they have not worked out. Thus, experimental biology has really provided us with a number of new tools to produce vaccines by many different methods. We expect that there will be additional vaccines made against even other infectious agents, such as respiratory syncytial virus vaccines, which are now experimental, and vaccines against other important causes of disease of mankind.